Consciousness, memory, and identity are emergent properties of a highly ordered biological network. Think of the human brain like a sprawling power grid. It operates on 86 billion neurons communicating across trillions of synapses. When this system begins to fail, the nodes flicker and die out in a catastrophic wave of structural collapse. Alzheimer's disease is exactly that kind of network failure. We often discuss it as a fading of the mind, but in reality, it is a relentless physical destruction of the brain's architecture. You can see the devastation with the naked eye. In a diseased brain, the neural circuits are dismantled, synaptic connections are destroyed, the cortical tissue volume severely decreases, and the organ literally atrophies, leaving behind deep, hollowed-out sulci. This structural decay takes root decades before a patient presents with mild cognitive impairment, or MCI. By the time severe dementia is clinically diagnosed, the biological damage is already massive. Yet, this large-scale organ failure isn't caused by a virus or a macrotrauma. It traces back to a single microscopic event, a thermodynamic error in protein folding. Outside the neuron, floating in the extracellular fluid, is a ribbon-like protein called an amyloid beta monomer. In a healthy brain, these proteins are continuously produced and dynamically cleared away without issue. The disease initiates when these normal proteins undergo a specific conformational change. Under certain chemical stressors, the monomer misfolds, exposing hydrophobic or water-repelling regions along its structure. Because these hydrophobic regions naturally repel the surrounding aqueous fluid, the misfolded protein is forced into a highly unfavorable energy state. To stabilize and hide these exposed regions from the water, the individual monomers stick together. This exact mechanism, where individual misfolded monomers first bond into unstable clusters, is known as primary nucleation. It is a simple, highly localized deviation in protein thermodynamics. But this minute error initiates the entire disease cascade, eventually bringing down the macroscopic cognitive network. For decades, the medical field focused heavily on the large, visible amyloid clumps. But to understand the true toxicity, we must look at molecular kinetics. This diagram isolates the two distinct stages of aggregation. On one side, you have highly chaotic, shifting intermediate clusters called oligomers. On the other side, you have structurally static, heavily stacked geometric sheets called fibrils and plaques. It is the highly dynamic oligomers that cause the active damage. Because their shapes are constantly shifting, these transient clusters readily bind to sensitive neuronal receptors on the cell surface, fatally disrupting the intracellular calcium homeostasis. Over time, these unstable oligomers continue to aggregate, elongating and settling into mature beta sheet structures. At this stage, they become structurally static, inert, and stop actively disrupting the cell membrane. This forces a major correction to how we view the pathology. The large, mature plaques found in autopsies are largely dormant. They are the tombstones of the disease. The transient, shifting oligomers are the assassins. Therefore, any effective pharmacological intervention cannot rely on clearing away the dormant tombstones. It must intercept the active, dynamic kinetics of the oligomer phase before the cell is irreparably damaged. While amyloid attacks the cell from the outside, a second structural failure occurs inside the neuron. The internal architecture of the cell relies on a network of microtubules, essentially a vast transport highway system that delivers nutrients. This highway is clamped together and stabilized by pristine tau proteins. The toxic extracellular disruption caused by amyloid oligomers triggers a reaction inside the cell. It activates specific kinases, which begin hyperphosphorylating the tau proteins. The rapid addition of bulky phosphate groups forces tau proteins into a massive conformational shift. The clamps spring loose, detaching from the microtubule entirely. Without the stabilizing tau, the transport highways completely shatter. The detached hyperphosphorylated tau then misfolds into dense, insoluble neurofibrillary tangles. Cut off from its nutrient supply, the neuron inevitably undergoes apoptosis, or programmed cell death. The entire pathological model becomes clear. A simple extracellular thermodynamic error dictates an inevitable cascading intracellular structural collapse. Historically, our ability to measure this structural decay was severely limited. Diagnostics largely meant waiting for late-stage subjective cognitive tests or confirming the damage post-mortem. This scan demonstrates positron emission tomography, or PET imaging. This was our first major diagnostic breakthrough, allowing us to accurately map the localized burden of amyloid and tau in living tissue. However, deploying radioactive tracers and complex hardware at scale remains structurally difficult and extremely costly. We are now driving a new analytical revolution, the shift to ultra-sensitive blood-based biomarkers. By extracting REL plasma samples and running them through highly automated precision immunoassays, we can track the disease in real time. These instruments are hunting for incredibly low abundance targets circulating in the bloodstream, specifically isolating exact molecular fragments, like plasma p-tau217 and the A-beta 42/40 ratio. This graph plots how we validate these immunoassays. To reliably detect targets at such minute concentrations, biochemists have to push the metrological limits of the hardware, strictly driving down the limit of blank, the limit of detection, and the limit of quantitation. By achieving this strict clinical linearity and maintaining tight coefficients of variation across highly diverse patient populations, we can confidently move these diagnostics out of centralized reference labs and directly into accessible point-of-care screening. Alzheimer's is no longer an invisible pathology. Through validated assays, the medical field now treats the disease as a quantifiable, trackable data stream, and with precise data, we can finally deploy precise interventions. The latest FDA-approved therapeutics aren't simple, blunt, small molecules. Drugs like lecanemab, pictured here, are massive, highly specific IgG1 monoclonal antibodies. This class of antibody utilizes a conformational epitope to achieve its target specificity. These proteins are engineered to recognize three-dimensional shapes rather than just chemical sequences. Lecanemab ignores individual monomers and dormant plaques. Instead, its variable regions perfectly lock onto the specific jagged geometry of the toxic protofibrils. By aggressively binding to these intermediate structures, the antibody halts their progression, deliberately steering the biochemical equilibrium away from further oligomer buildup. Once bound, the antibody triggers a secondary response. The antibody's base acts as a flag, signaling the brain's resident immune cells, the microglia, to swarm the area, engulf the tagged complexes, and clear the toxic debris from the extracellular space. We are not chemically altering the brain with broad inhibitors. We are deliberately teaching its own immune system to identify and correct a microscopic thermodynamic error. But pharmaceutical intervention is only half of the equation. The human body possesses a powerful secondary defense mechanism known as cognitive reserve, driven by neuroplasticity. Rigorous cardiovascular activity provides the mechanical stressor required to trigger the expression of brain-derived neurotropic factor, or BDNF. BDNF is highly neuroprotective. It promotes the survival of the brain's existing neurons, while actively driving the growth of dense new dendritic spines to bypass damaged circuitry. Coupling this with strict systemic metabolic management, specifically minimizing insulin resistance through diet, drastically reduces the chronic neuroinflammation that otherwise accelerates tau pathology. These are lifestyle interventions that actively construct a biochemical environment wholly hostile to neuronal decay. By mapping the exact molecular kinetics of protein misfolding, pushing the analytical limits of blood-based immunoassays, and deploying conformation-specific antibodies, the medical field has reversed decades of stagnation. We are no longer passively observing the catastrophic structural collapse of the brain. We are actively and precisely engineering its defense.